Good afternoon, ladies and gentlemen. Namaskara. On behalf of the School of Life Sciences and the Indian Academy of Sciences, it gives me great pleasure to welcome all of you to this very important discourse today. At the onset, may I request our distinguished guests, Professor Spyros ARTAVANIS-TSAKONAS, Professor Vidyanand Nanjundaya, and the Pro-Vice Chancellor of St. Joseph's University, Professor Ronald Mascarenhas, to kindly occupy the seats on the dais. Thank you. We begin today's proceedings with the lighting of the lamp, an ancient tradition in our country, to mark auspicious beginnings, and symbolizes the triumph of light over darkness, knowledge over ignorance, and invoking God's blessings. I request our esteemed guests to please come forward to light the ceremonial lamp. It is now my privilege to extend a formal welcome to our guests and participants. We deem it an absolute honor to host at St. Joseph's University today, Professor Spyros ARTAVANIS-TSAKONAS, Professor Emeritus of Cell Biology at the Harvard Medical School, and the Academy's RAMAN Chair Professor. We recognize that this is a rare and special privilege. This gift, this opportunity to be in the same room and listen to someone who has done cutting-edge scientific research for many years, from fundamental elucidation of the Notch signaling pathway through building the first large-scale protein interaction map of a complex organism and linking these to development, cancer, and neurodegeneration. And this experience can therefore gives Professor Spyros therefore the ability to zoom out and talk about this big picture in biology. Professor Spyros, it is truly an honor, and we are genuinely excited to have you here today and to listen from you. I now request our Pro-Vice Chancellor, Professor Ronald Mascarenhas, and Professor Vidyanand Nanjundaya to honor our guest with a token of our deep gratitude and appreciation. Thank you. Welcome. Welcome.
Thank you. So,
we also very gratefully acknowledge the presence of Professor Vidyanand Nanjundaya, Professor at the Center of Human Genetics, Bangalore. We thank you for taking the time out of your schedule and being with us today. Professor Ronald Mascarenhas, no, we just say we're continuing with the welcome. Thank you very much for being here.
thank you.
Yes, absolutely. Professor Nanjundaya, thank you for your presence today. We're very honored that you're here. Professor Ronald Mascarenhas, our Pro-Vice Chancellor, has always been very supportive of everything that we do at the School of Life Sciences. So, thank you. Dr. Sujata, Dr. Mahesh Chandra, and all the team from the Indian Academy of Sciences, we acknowledge your presence. Thank you for being here. All faculty and students from St. Joseph's and neighboring institutions, welcome. We have no doubt that this will be an insightful session and that you will take away understanding and fresh perspective. Again, a very warm welcome to everyone here today. And I now humbly request Professor Nanjan Daya to do the formal introduction of our speaker.
Thank you very much for this opportunity to introduce someone who has made a remarkable name for himself in modern biology. There's a lot that can be said about him and his work, but since we've all gathered here to listen to him and not to me, I'm going to keep myself brief. Professor Spyros Artavanis is of Greek origin. Like the hero, or at least one of the heroes of his talk today, Aristotle, he had his early education in Greece. He had his early education in Greece and followed that up with an undergraduate degree in the University of Cambridge, where, as he just informed me, he was surrounded by a galaxy of the great people of the time. Following that, he studied as a postdoctoral fellow, studied and researched as a postdoctoral fellow, again in institutions of the highest renown. I put things the other way around. So he did his degree, if I remember right, in ETH in Zurich, and then went to do further work in Cambridge, in Stanford, and in Yale University, and finally ended up at Harvard University, where he is now. He was one of a small number of people at the time, in the mid-1970s, who pioneered the area that's now, well, roughly called molecular developmental biology or molecular genetics. So basically the idea being, how do you tear down, in a sense, a living creature to its most fundamental genetic constituents, and then figure out how those constituents work with each other to actually give you the creature. The second part is much more difficult than the first, but that's the one that developmental biology is actively embarked on today. He's a person, as I said, who has made a great name for himself in his field, and like all of you, I'm looking forward to his talk. The title promises a great deal. It says from Aristotle to AlphaFold. Aristotle is known to us in this country through one of his students, Alexander, about whom all of us heard as perhaps the first real conqueror of North India. Following that, of course, we remember Aristotle as standing at the head of the scientific lineage which has given us what we call modern science today. And it's interesting that this lineage is in parallel with other lineages which also contributed significantly to modern science. The lineage starting from Aristotle, of course, had a somewhat tortuous history through Islamic civilization and then what's now called Western civilization or Europe, and now it's become worldwide. The Indian and Chinese traditions, which are the other two well-recorded traditions of science, either have had or have not had links to the present. This is a matter of some discussion among students of science. Now, Aristotle is famous for another reason, or should be famous for another reason among us, which is that his tradition of doing science involved actually going out and looking at things and actually doing, in some primitive sense, experiments, though they are better called observations. This was rather different from the purely observation and analytical school of traditional Indian philosophy, except of course for the Lokayata school, which had a firmly materialistic view of nature, not very dissimilar to the view that we have today. The other point about interest with regard to Aristotle, and that also figures in the abstract of Professor ARTAVANIS' talk, is his use of the word pneuma, whose translation I will not attempt, I'm not very sure, but it approximates roughly to what are called the doshas in our tradition, very roughly a cross between breath or prana in a rough sense and the doshas. So there's really a lot to look forward to what's going to come in this talk, and he has promised to take us from Aristotle to the alpha fold, which is essentially the science not just of now, but you might say almost of this very moment. And for those who have not heard of it, this is this huge advance in artificial intelligence methodology, which was started by Google as an offshoot of what Google called deep mind. And it's led to a revolution in terms of predicting structures of proteins. So there's a huge jump from Aristotle to AlphaFold, and Professor Spyros Artavanis-TSAKONAS is going to take us through across that stretch. So I welcome you once again and look forward eagerly to what you have to tell us. Professor Spyros.
The public lecture was very fun and extraordinarily dangerous because it's very difficult to try and find the balance in the text that will allow me to tell you what I was saying. I think also that it's really children moving that we start from the So what I was saying is that Dennis Hasabi, who was the principal in DeepMind, well, he's not Greek, but he's half Greek. His father is Greek, so we start with a Greek, at least now, and we will hopefully end up with a Greek, assuming I will have all the time that I need to tell you what I wish. So biology is, as I said in a little abstract, is the science that is closest to us. The science that's three-dimensional, as opposed to physics, which has n dimensions, and it's much more difficult to understand intuitively. And biology, if you don't understand something that I explain or anybody explains, it's my fault, not yours, because the concepts are fairly straightforward. As I said, they are three-dimensional. So we will have a trip, I hope, a pleasant one, that brings us from Aristotle and 2,000 years later. So it's a trip that will bring us about 2,000 years all the way to today, hopefully. So the beginning, and as already was mentioned, the contribution in science of the Chinese or the Indians, I am not going to touch that. I will be talking only about Western, rightly or wrongly. All right. I mean, I know that Chinese, for example, had quite a lot they had a lot of biology, let's say, early on. So the beginnings are considered to be called the Greek period, which is pre-Socratic, and then eventually Aristotle. So he has been sort of a landmark. The pre-Socratics, that's before Aristotle really, they are actually quite close to us as scientists. They have it is the period between the 7th and the 5th centuries, and it is defined by a shift from mythology to actual rational explanation. And it is the pre-Socratics, they are essentially using the same methodology that we use, reason, discourse, and observation, to understand the natural world. They don't rely on myths and necessarily metaphysical explanation. So perhaps the most well-known pre-Socratic is Democritus, and he had a very interesting and very modern way of looking at things. What he was arguing, what he argued, was that there is void, so there is space, that there is nothing there, but this void is full of elements that they come together. They call these elements atoms, all right, a word we use today, and they come together to construct, if you will, the world we perceive. Imagine having that and coming these molecules are coming, these atoms coming together and making a chair or a person or something. Okay. A rather modern concept, if you will. Now, Democritus was one of these pre-Socratics, but there are others who relied not on atoms, but on other kind of elements to explain the primary substance, because that's something that we're still looking for, if you will, in many different ways. So I'll go through them in just a little bit. Thales thought that water is the primary substance. Anaximenes was air. Empedocles was earth, water, air, and fire, a concept that is familiar to you in India for sure. But then they started sort of thinking a little bit more abstract. And Anaximandros, for them, the substance was not a substance, but it was the infinite. An interesting concept. Something that has no end, and I'll come back to that, has no end and has no beginning. And the Pythagoreans, they were the most esoteric, if I may say so, of all, because they felt that everything are numerical relationships. Again, a very abstract kind of thought, but one that was actually quite influential. Very good mathematician, if you will, all right? So, according to the pre-Socratic thought, every physical phenomenon that we perceive, that we see, however complex, is the result of interactions between simple elements. That's a concept that we should keep in mind. It's very, very, very important. And the creating principle is what it's called, it's the automaton. It's called the mechanism, or the mechanism. And basically, what it means is chance. So you have things that meet each other, they build something that is useful, if you will, and that thing stays. And I'll come back to that concept. However, the world of the pre-Socratic is one that has a beginning and an end, which means that who made the beginning? And like so often, even today, they postulated a beginning that they postulated a creator, somebody metaphysical who actually started everything, all right? How it went afterwards is a different story, but somebody had to start it. If you have something that has a beginning and an end. We go to Aristotle, however, which comes after the Socratics, the pre-Socratics, He hated the pre-Socratics. He didn't like them. He felt this notion of chance and mechanism and so on was not right. It was wrong. And he started sort of developing theories that were almost on purpose, purposely sort of thought out to contradict the pre-Socratics, right? So, but he did something which is very, very interesting philosophically. He invented, essentially, a universe to get rid of all this, the notion that you need a creator, and a universe that is infinite. There's no beginning, there's no end. So you don't need to have somebody to start the beginning. An abstract, but quite an interesting sort of a philosophical thought. The biology, however, in his world, is teleological, and I'll explain what I mean by that. It is the final outcome that provides a causal explanation of the phenomenon, all right? So the reason the reason teeth develop is because you need them to eat, all right? And I'll contrast that with the pre-Socratic in a second. He develops, again, as I say, teleology against the pre-Socratic thought of automaton, of mechanism, chance. The pre-Socratics are chance and necessity. I mean, really. I mean, you have, by chance, something comes together, and then it stays because it is useful. An extremely Darwinian attitude. I'll come back to that. So the pre-Socratics are actually closer to us in many different ways. I argue, and it's not only me, that the pre-Socratics were Darwinians, or that Darwin was pre-Socratic. And why do I say that? Because their argument is that teeth developed. Remember what Aristotle said. But they will say they develop, and then by chance, since things come together, they develop teeth, and they stay on because they are useful for survival. 100% a Darwinian thought. So, and indeed, it is the chance and the necessity. So you have by chance to create something, and it is the necessity because it's useful that it stays on during evolution, all right? Now, however, in spite of the wrong notion of teleology, Aristotle contributed an enormous amount, and you actually mentioned, hinted to some. He sets up the basis of a methodological, it's a Greek word too, I can't remember, of a methodological approach, and that for experimental science. And this is the first time that something like that has been done. And he uses essentially what we use today, and a deductive and inductive approach to reach conclusion. He doesn't reach a conclusion and then try to find the proof. That's the other way around. He looks, he observes, and then he reaches a conclusion. Now, one of the things that he did, there's this famous book of Aristotle about animals. It's worth looking at it, especially if you are students and you are starting your career. It's useful. It's useful. He's essentially the first to look at embryos, right? He's the first embryologist by any means. And what he did was he looked at chick embryos, and he described that the development of the chick embryo is gradual, an important concept which we will get back to later. So that you start with an amorphous juice, if you will, all right? And then this develops to get the chick as we know it. And the notion, however, of teleology remains almost 2,000 years. I mean, Aristotle is gone, and the teleology is around for a very, very, very long time. So it is arguably that that Aristotle is one of the most influential biologists ever. 2,000 years is not too bad. I don't think anybody will remember my papers for another 3 years, let alone 2,000. All right. So his biology, as I said, remains until the 17th century, but and 600 years of Greek tradition disappears. The Romans don't accept, don't adopt, rather, I should say, biological thought. Nothing is happening. And essentially, biological thought starts again by the hero, for me, of all times, and that's Leonardo da Vinci. It is Leonardo who starts looking again at the biological world in a way that has not been looked at before. So he's doing comparative anatomy, observation indeed, no experiments, that's for sure, and looks at essentially how the correlation, anatomy that is functionally important. Remarkably, Leonardo da Vinci dies, nothing after that. It takes 50 years before this is again put into place. And it is Bellon, a French biologist, who started doing comparative analysis of, in this case, birds and humans, 50 years later. So, as usual, Leonardo da Vinci, way ahead of his time. Now, in order to understand biology, however, we have to develop the concept of cells. Biology without cells is no biology, all right? So we'll spend some time talking about that, about cell theory. And in the 17th century, a remarkable thing happened, and that is that Zacharias Jensen, a Dutch, actually, he was a lens maker. He discovered a way to make a 20x microscope. Now, I'm saying that for two reasons. One is because that was a remarkable achievement for biology, but also to emphasize that in biology, we are depending on new technology. So it is the new technologies that will make the conceptual leaps, really. Whether we like that or not. We're not as smart as the theoretical physicists. So I think the conceptually important jumps are associated with new technologies. So he discovers a 20x microscope. So you can imagine you start looking at everything. You have something that you look at your hand, and you look at it under 20x microscope, you see all kinds of different things that you didn't imagine were there. So that, of course, is it's a huge advance, because people started looking at the natural world with, and there are three characters that are very important in the development of cell concept, if you will, as we will see. The first one is Malpighi, Marcello Malpighi. He's an Italian physician. The second one is Anthony van Leeuwenhoek, who was a Dutch scientist. And the third one who may be the most important scientist of the 17th century, is Robert Hooke. Now, I don't have a mugshot of him, but the reason is actually quite interesting and very modern. He had a dispute, scientific one, with Sir Isaac Newton. Apparently, Sir Isaac Newton was not a stupid person, obviously, but a very vindictive guy. And in the book that Newton wrote about light, Hooke said that whatever is right in this book, he stole it from me, and whatever is original is wrong. So that really, I mean, that made Sir Isaac Newton extremely angry, and he wiped out all references to everything that had to do with Hooke. And to that day, we don't have a portrait of him, or however, in this the book of Hooke, the Micrographia, which you can still find it even on the internet. Actually, you can see it. It's a remarkable book on describing the natural world. Really, really remarkable, right? Now, the microscopes allowed the people using them to start seeing structures that they couldn't quite understand. And the structures that they couldn't understand, but they saw that everywhere, are the sort of globules. They were round things that Hooke said that bubbles surrounded by a halo. Nucleus, a cell, and the cytoplasm are surrounding it. Didn't know what this was, but he saw that in blood, and he also saw that in plants. And the plants have thick walls, so that was very easy to see. Whereas in mammalian system, so Hooke, as I said, saw these bubbles, and they were looking at plant cells, which have this very thick wall around them, so they were easy to see. The mammalian cells, or the metazoan, let's say, cells, they were not easy because the microscopes were not good enough to see the cell membrane, let's say, let me put it that way, all right? But there were a few things that were extremely important already at that time. The one is that Hooke saw this repeated structure seen in many different plants. So conservation of that structure, maybe this is a quantum. This is something that these bubbles or these round things is some kind of a basic structural element. And Leeuwenhoek saw actually corpuscles, corpuscles in the blood. And neither of them, however, realized that these are cells. And for almost 150 years, scientists were chasing the globules, trying to figure them out, trying to understand them, trying to see how they differ from species to species, for animals. So for almost 150 years. It is the lack of thick walls, as I said, in plants that prevented comparison between flies. That's a Freudian slip, I'm sorry. Between plants and animals, all right? So as we advance, however, and make better microscopes, things are becoming clearer, literally, all right? The microscope that corrected spherical aberration allowed us to see things in a much, much, much better way. And that, of course, led to the Trochet, who are now in the 19th century, early 19th century, who says that nature possesses a uniform plan for the ultimate structure of organized being, both animal and vegetable. An absolutely remarkable statement that nature has a blueprint that has commonalities among species. So there's conservation, and conservation is something that I will be hammering from now on quite a bit, because that's what it's all about, really, as far as I'm concerned. The people that developed most of the cell theory was Matthias Schleiden and Theodor Schwann. This is now 19th century, early 19th century, essentially, right? And a colleague of theirs, Virchow, the question that arose with, I mean, everybody accepted cells, all right, but how do they arise? How do cells arise? And what was realized was that cells arise by division of pre-existing cells. Again, a fundamental concept. And it was this Remak and Virchow around middle of 19th century that realized that. So from one cell you have two, from two, four, and blah, blah, all right? And Schwann is making this, again, I will say it again, profound statement, that all organisms are composed of like parts. Absolutely an amazing conclusion, all right? So by the second half of the 19th century now, the cell has a nucleus, it has a cytoplasm, it has that has a nucleus that's closed in the cytoplasm, it has a membrane outside, and these are conserved parts in all animals, all right, in all living organisms. So what about development? We know how cells are done. We know how they are multiplying. We know that they have some particular structure, and we know that everything is made out of cells. Ontogeny. Now that's where a lot of fun comes in, right? Because in order to we have to explain how development works. Yes, they all are cells, but how do they come together, right? And it is Cartesian thought, the Renaissance, that essentially challenges finally the Aristotelian thesis, which to an extreme, to an extreme, it transforms organisms to machines that you are built up in a very precise way, like a machine, right? The protagonists of the scientific revolution were, however, again, people of faith. So we go back to the pre-Socratic notion of God, if you will, all right, of metaphysical, and God remains for them the ultimate architect of the universal laws, and it is on us, as scientists, to discover those laws. But the laws were set by something else. So, as I said, the pre-Aristotelian universe returns, and the so-called physicalists, which are the pre-Socratics, really. I mean, the people who are interested in explaining phenomena mechanistically, really. That inspires, of course, like everything else, you know, an antithesis, a movement that is different from that, just contradicting this particular thesis. And that was so-called vitalistic movement. So the vitalistic movement provokes another physicalist movement, and they were fighting how things are done, whether it's metaphysical, forces or not. There's a huge fight for decades, right? And indeed, this is distilled into two things. One is when you look at development, some people, like Aristotle indeed, right, remember the first embryology, says that the embryo starts as a formless egg, there's no form, and then slowly it develops into a formed organism. The preformationists, right, they actually say that the development is the unfolding of existing structure. There's one famous such notion, the homunculus. So the idea of the homunculus, even though in 1651 Harvey confirms Aristotle's gradual development, and it is the gradual that I am harping now more than anything else. All right, it's progressive. Development is progressive. All right, in spite of the fact that this is 1651, right? Nevertheless, the preformationists come back, ignore that or don't understand it. I don't know, and insist on this homunculus. So, what is a homunculus? The so-called spermatists, people who thought that sperm is the origin of everything. They were the counteract that egg is the. It's like like religion striking. I mean, possibly. So according to this dogma, you can see now this is a sperm, a thing of a sperm, and in the sperm there's this little human, tiny little human, tiny, but it's formed. It has legs, it has head, it has hands, it has heart, it has everything. And as development proceeds, these things are becoming bigger and bigger, and until it makes you. All right. So, which doesn't make very much sense, but nevertheless, I mean, this is that is really the thesis. And a twist of this is the so-called recapitulation, which this is a notion that comes from the German Romanticism, really, and it's that everything in life goes from low to high. So there are low organisms and high organisms and everything in between. All right, and there is this famous one of the heroes of that notion is Haeckel. And recapitulation, of course, has notions, has connotations of racism as well. Because as you go from low to high, blue people may be lesser than green people, like black people, like white people, and so on. And that has been used by throughout the 20th century as an excuse to do all kinds of things. I'm sure that Donald Trump is a big fan of Haeckel, but the problem is that this is of course complete nonsense. All right, this famous figure that you have development from fish all the way to human on the right, and the idea of recapitulation is that the development of a human, for example, all right, it goes as it develops, it goes slowly through. It recapitulates phylogeny. So you have an early organism, and you go on. And in fact, they were claiming that there are embryonic, there are proof, experimental proof of that, and that is they were getting it from the French teratologists who were looking at human abortions, random abortions, and this is one that was published, and on top there's a human fetus. On the bottom there is a clam, and they claim that this human abortion is close to the clam. I mean, it just didn't hold, all right? Not only it didn't hold, but before Haeckel's time, Karl Ernst von Baer, one of the biggest intellects of the 19th century, he said that the malformations, correctly, may represent arrests at specific developmental stages, but they do not represent the adult forms of existing animals. This is not, it was, in spite of that, Mr. Haeckel, here he is, he completely ignored that, or he didn't know about it, I don't know, because he came afterwards, and he developed his recapitulation theory. But if you think about it, the epigeneticists and the other school who believed in God and so on, there is a vital force that actually exists. It's not metaphysical, but we call it genome these days. So you start with an egg that has all the information in there, and the sperm, mind you, right? That has all the information to make a homunculus, to make a human, all right? So that's something which is clear today, all right? So what about genetics and heredity? We know that, or they knew, that sperm and egg are cells. Cells, we know that cells don't arise de novo, but they derive from pre-existing cells. And this is an unbroken series that people thought at the time even, that this is what heredity is all about. And germ cells constitute sort of the physical basis of heredity. Hertwig, we already talked about that. He mentioned that the cell nucleus is that what carries the physical basis, and von Benetton connects chromosome behavior with heredity again. All right. So heredity and cytology are being linked. Mendel, he's late 19th century really. He discovers genes, genetics, and how these genes behave, but he remains completely unknown, obscure. I mean, he's a monk in some Russian God knows where. They just don't, nobody knows about him. And it takes another 15, 20 years until Mendel is rediscovered. Oops. By De Vries, Correns, and Chermak, really, all right? And it is T.H. Morgan, who used to be an embryologist, actually, originally, and his magical group of students in Columbia University. You should actually look this up, because these were a strange group of people. Even a communist was there. And everything was very, 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 it must, I mean, if you ask me, what time do you want to go back to and live in? It would be the Morgan's Lab. An extraordinary place. A lot of them got Nobel Prizes later on. And they have been looking at mutations. They've been isolating mutations in flies. Why flies? Because they are easy to work with, basically, all right? However, the genes that geneticists were studying were not thought to have anything to do with morphogenesis. Again, a very important sort of distinction. And there was a big fight between geneticists who they thought that these weird people, the sort of hippies of the time, that had nothing to do with morphogenesis per se. And there was a huge hostility, actually, between the embryologists from the early 30s until the middle 40s, between the embryologists and the geneticists. So Morgan has been isolating, as I said, mutations, and this is one particular mutation that has notches in the wing. Sujata is smiling because she knows that this protein or this gene has been my life for 40 years. But I want to show you, the reason I say that is to give you an example of what's going on. So you have the wing of the fly has these little notches. So the embryologist said, fine, I accept the wing that it has notches, that this is a mutation, but what on earth does that have to do with morphogenesis and embryonic development and the patterning of them? Big controversy, early 30s, two people, Salome Gluckson Welch, looking at the T locus of mice. She connected mutations with embryonic phenotypes. And Don Poulsen, who was a fly embryologist, he was linking pieces of chromosome with specific mutations, deleting pieces of chromosome. And he actually is the one that found that in a particular band in the chromosome 3C7, that's where notch is. And he linked notch loss of function with a particular phenotype. Now, biology, this is now middle 30s. Biology moves fast. DNA is identified as a genetic material. The elucidation of the DNA structure by Watson and Crick is happening, and the molecular revolution starts being there. So, by the 60s, genetic code is there, operon concept has been developed. We know that genes can be switched on and off, which explains to some degree why or how, possibly, the same genome from cell to cell can express different proteins because of these switches, the operon concept. Of course, yet how genes combine their action to drive development remains a problem. Okay? So I want you to think of something very basic. Imagine that you are constructing a three-dimensional organism, a fly, you, me, a horse, whatever. The cells, in order to build that organism, it is intuitively obvious that you need cell communication. Cells have to communicate so that, the red cell has to go close to the white cell, the white to the blue, and so on. So you make something that makes sense and is functional, all right? Now, if I also told you, we all agree on that. I think that doesn't, that if you look at the genome of the fly and the genome of the worm and the genome of us, it is essentially the same. Now, I'm taking sort of poetic license here to say the same, because they're not the same, but the basic genes are conserved. And clearly, the humans and worms are not exactly the same, all right? They have different shapes and so on and so forth. And if I were to tell you also that the signaling pathways, the cell communication pathways that exist are not very many, are a handful, and these signaling pathways are exactly the signaling framework, let me put it that way now, is exactly the same between flies and humans. Why not? All right. These pathways are pathways that many of you have heard, even in your undergraduate classes. There's the Wnt pathway, the receptor serine kinase, hedgehog, etc., and the notch signaling pathway, which, as I alluded to, has been my bread and butter for a very long time. These pathways have been evolved in the Precambrian. So the biggest problem for me in biology now is how is it possible to have the same signaling framework, and within that framework you invent novelty. Evolution invents novelty. So you start with something that is low, if you will, all right? And it is the same pathway that will allow you to make a hand, a liver, or whatever, instead of a wing. So that is a profound problem. We don't know how that works. And it is actually also quite clear that these pathways are extremely complicated. I will finish this last half hour. No, I'm joking. With telling you something about complexity and finishing up with my friend Demis Hassabis, the other Greek at the other end of the story. So, if you take notch, that we are not familiar with notch, all right, and we know that notch has these wings, these notches on the wing. So what you can do, you can ask a very a simple question. The answer is not simple, but the question is, you can ask how many genes, can I find genes in the genome that will actually modify this phenotype. So this is a loss of function phenotype, never mind, it's a loss of function phenotype. And I can take this phenotype, this genotype rather, and combine it with other mutations systematically, every single gene in the genome, because we can do these things now. Omics and what have you, all right? We can do them. And ask the question, can I find suppressors and enhancers of this phenotype? And sure enough, I can. I can find both suppressors of that phenotype and also enhancers which make the phenotype work. If you ask the question, how many of those can you find? And in this particular screen, we screened 50% of the genome. This is quite a work, I mean, it's not trivial. This is approximately 500 genes. Confounding complexity. Drives you crazy. You feel like quitting. You don't know what to do next, because the whole object of the exercise is to be able to deconvolute this kind of complexity. But there is no doubt that the underlying, the genes that are capable of modulating notch signaling activity are in the hundreds. What do you do? So we decided to go ahead and define protein-protein interaction. So if you have two genes that interact with each other, like this guy here, you make it worse, or you make it better, right? Per definition, these two genes interact genetically. Functionally, that's significant. How does that happen? At a molecular level, it's a different story. And a genetic interaction, while true, no doubt about that, does not necessarily mean a protein-protein interaction or something that is direct. It can be through New York and Mumbai or something, all right? But they still, the bottom line is that it modulates the signal. And if I told you that modulating notch signals can cause cancer, that's important. And other diseases, by the way, all right? So what we decided to do was to take the cDNAs, clones of 15,000 genes that in the fly, take the cDNA, tag it, and I'll keep there. If you don't understand, I'll be happy to explain it later. So you put in a hook into that gene, and express it in cells, and see what else interacts with your cDNA. And then analyze that in mass spec. And that will define protein-protein interactions, and then classify those in complexes, specific complexes, all right? Because a protein in a cell never acts alone. It acts in concert with other things. And so this is not a solution. It's a map that defines problems rather than solving them, but it's a start to try and deconvolute this complexity. So this here, and I will go through it in excruciating detail. These colors are complexes that are actually are defining in ontology. They are defining similar functions for, and you can actually see that all the maps that we have made in flies were defined by about 600 complexes. Why is this important? Well, two things. One, this is a blow up of this in one. So the idea is that this complex here, you can actually first of all, does it have predictive value? If I take this complex, can I really say that if this complex has a notch element in it, a notch modifier, are the others also modifying notch? The answer is they have predictive value. About 50–60% of what we've looked at, it has predictive value, which is useful because a map without predictive value is not a map. All right. And so we are interested in asking, do complexes have predictive value? And I said yes. If we do AlphaFold, now we can do these things. So we can look at that in 3 dimensions using Dennis Hassabis' AlphaFold prediction. Do I see something special or different or whatever? What happens if I destroy one complex? Because now you look at the whole cell as a system, if you will, all right? So if I destroy one complex, do next door neighbors are affected? If I change the quantity of that, is it affected? So there are an enormous number of questions that you can ask. The AlphaFold I told you. Most importantly, do these complexes, are they conserved in evolution? So Wade Harper and Steve Gigi, 2 close colleagues at Harvard, they have done most of the work, have defined the human proteome. So what we can, I mean, I'm obviously not going to go into details, but we can take a complex from a fly or the other way around and ask the question, does this complex is still a complex in humans? And conservation in general, we take it as a sign of importance, biological importance. in complex operation. Is there now an integration code? Is it the pathways that talk to each other? Because remember, we have 7 pathways, and we have worms all the way to humans. That's it. So, is there a code in talking in integration of signals that is conserved? And above that code, you can add on different elements so to make it more complex and evolve species. Because evolution, per definition, has to have the means and given the possibilities to evolve new structures, new functions, right? Same thing. So these are questions that I feel the most important one right now in biology. Demis Hassabis and is right there. And the AlphaFold, this kind of technology gives us a very different perspective. It's like Janssen's microscope. It's an analogous thing. I mean, this is a new way of looking at things. And omics has given us this extraordinary capability of generating data in a mindless way, if you will, all right? Nevertheless, a machine learning and AI technology will allow us to try at least and deconvolute and look at relationships in this sea of information. One always, artificial intelligence, everything, you go to the grocery store and something is artificial intelligence, AI, which is getting very painful at the end, and because nobody knows what it is, they don't know how to use it, etc. However, do I think that that AI can help us address complexity, which is in biology is extremely complex, whether we like it or we don't like it. And can AI help humanity? My view, which I'm happy to defend and debate, is yes. So I thank you very much for your attention, and I'm happy to answer questions.
Questions? If there are any burning questions, hands up. We won't have a lot of time, but are there any questions from students? Yes?
No, it's okay. Good afternoon, sir.
Hello.
This might be a basic question, but I wanted to ask how the cDNA that you mentioned interacts with what other components or like what if genes. Could you elaborate a bit?
I have to repeat the question. I wasn't clear what I
Okay, sir. So cDNA that you mentioned,
DNA.
yeah, you
Yes.
mentioned if you tag it and how it interacts with other components.
Yes.
I would like to know more about how it interacts with other genes,
Yes.
if possible.
Well, I mean, when you express a particular protein in a cell, and the protein does not act alone, but it acts together synergistically with other proteins, you can define these complexes by mass spec analysis. And that's how we do it. So you tag, you put a hook, in other words, all right? You throw it into the cell, express it, and then you isolate with the bait, or rather you put a bait, and you isolate with the hook the complex. Now, it has many problems. The whole approach has problems, but it's a start. And for example, one problem, an interesting one, is this represents one cell type. What happens in other cells? Is everything the same? And there are differences. Steve Gigi and Wade Harper have done if you look at, I mean, you look at that, so it's published. They looked at the different cell types, and there are differences, interesting differences. And you expect that. I mean, you know, a blood cell will not have the same proteome interactome than a liver cell or a neuron.
Hello. So Drosophila has been a central model for our understanding of many various cell processes.
Yes.
How do you think like the model fits with the understanding of neurodegenerative diseases in humans? Like in what ways?
I'll answer indirectly, and you will see where I'm going. In the last 3 years, we have been working on neurodegeneration. We have been working on Parkinson, on ALS, on Alzheimer's. And of course, the guys don't have Parkinson, they don't have ALS, and they don't have Alzheimer's. But the basic biochemical pathways that modulate genes that are involved in disease, like FUS in TDP43, and PAC and PINK, and tau and A-beta. You can actually identify relations. Flies will not solve the problem. Flies will define a problem, and it will make your life easier in humans or in whatever to ask the right questions. But if I tell you that X interacts with Y in flies, you can say, all right, maybe that's the same thing happens in humans, and that could be important. All right, both, you know, I don't know what's happening. I think this is the atmosphere outside, you know, the pollution. Yeah. So I think, did I answer your question? Yeah. Yeah, okay. Thank you very much. Thank you very much for the photography. Thank you for saying my name. It happens extremely rarely. So much so that if somebody does it, I'm suspicious. So. Yeah.
I have a friend who's in Greece.
All right. Yeah. No, perfect. I mean, I'm amazed. Well, thank you. Thank you very much. Thank you very much.
Before you leave, I now have the very joyous task of saying thank you. I'm still slightly at a loss of words about how illuminating that talk was, because I think in our biology lessons, and faculty here will agree, that we start with cell theory and then jump to Mendel and then jump directly into DNA and its story. So this was exceedingly illuminating in terms of how we should look beyond to understand the philosophy of science and how it's evolved. And I also think it was extremely important because now we have the advantage of hindsight and we take microscopy for granted. But we recognize that microscopy at that point was the AlphaFold of that time. And therefore, I really appreciate how you say that technology drives biology forward. And thank you for those lessons. So it was intense across 2 millennia, but fantastic. So, Professor Spiros, you have a deep gratitude for elucidating this so effectively and also shining a light on people who took biology forward or understanding of life and biology forward. And for so succinctly saying that we right now have only a map of the questions and how this complexity, there's more questions than answers at this point. So thank you very much on behalf of the management, the faculty and students of St. Joseph's University. Thank you. Professor Nanjandaya, thank you very much for being here and setting the context for this talk. It was important for us to think and understand what is going to come, and you very elegantly said it. Thank you for your time today. In absentia, Professor Ronald had to leave for another meeting, so I record my gratitude to our Pro-Vice Chancellor, all faculty and students. Thank you for being here. I'm sure you leave with fresh perspective and understanding. Again, thank you very much, everybody, and have a lovely afternoon.